Could you briefly, in 2 sentences, tell us what you do in your lab?
Well, our lab is quite different from most other labs. When you say a lab, people have a certain sense of what goes on in a lab. Right? They imagine scientists trying to understand things, and that part of it is true about our lab as well. However, if you were to take a pure science place, and the Indian Institute of Science would be a good example, the approach to the understanding of a problem that is used at the Indian Institute of Science versus the way we work in our lab has many similarities but some important differences. First off, we are located on a medical college campus, and we started this activity from a very different motivation. When we started this work, we wanted to actually try and understand those aspects of the disease biology that made a difference to patient care. And one of the critical choices that we made when we started our work was to focus on the specimen, that is the human disease specimen itself, the breast cancer tissue. So my laboratory has, for the last 10 years, used as its central material, so to speak, the breast cancer specimen itself. The second part, which we are very different in our lab from almost all other labs, is that we wanted continuity of contact with the patient from the very beginning. So we did what in technical parlance is called a longitudinal cohort study, in which patients are enrolled into a study at the beginning of it, and then you stay in touch with them over long periods of time. So we have built a breast cancer cohort of 446 women who were diagnosed with breast cancer, and for the last 10 years my lab has been involved in staying in touch with these women as they went through their disease, as well as analyzing their specimens in our laboratory.
Are you aware of any other groups that are doing this kind of a study in India?
Yes, actually we are not the only group. In fact, I would say that almost all hospital-based research groups, be it the Adyar Cancer Institute or the Tata Memorial Hospital, have a very similar approach to research. And most importantly, I think this approach got a great fillip about 10 years ago, when the National Institutes of Health in the US set up what was called the TCGA. The TCGA stands for the Cancer Genome Atlas, and not coincidentally, those are the 4 bases that make up the DNA. And they approach this exactly the way we have structured our work, in that patients were recruited, their specimens were collected, they were followed up, and then these specimens were interrogated using the techniques and the tools of modern biology.
So how do you think this is significantly different from the kind of research that happens in a so-called lab? And how do you think it matters, especially in India?
The significant difference, I would say, comes from the typical motivations of a researcher in a pure science lab. There, most often the researcher is motivated by trying to understand how that process happens, so carcinogenesis itself, or how does the tumor behave, right? So they're propelled by trying to understand the fundamental biology of that condition. Whereas here, we were propelled to try and see if, based on the alterations we found in the breast cancer tissue of women who did well, quote unquote, meaning people who did not succumb to the disease versus people who unfortunately succumbed to the disease? Were there changes in their tumor? If we knew of these changes at the very beginning, they would inform the physician taking care of them, make better treatment decisions. So it was actually one of motivations, in short. We were motivated by trying to aid the treating physician make more appropriate decisions, and a fundamental lab research in a place like IISc is motivated by trying to understand the biology. Both are important, they're different, right? And in India, while both sorts of researchers and both sorts of researches have been done in the past, the uniqueness, if I were to say, of our approach was we attempted to and succeeded in staying in touch with exceedingly large proportion of the patients who were recruited, 96 to be precise. So that is 96% of all women who were enrolled into our cohort. We stayed in touch with them over a period starting from 2008 to 2017. To the best of my knowledge, that proportion, 96, is an unprecedented proportion for such a long time in the context of breast cancer in our country for these numbers.
I have this last question. I want you to tell people more of if people getting enrolled into such longitudinal research programs help treatment.
In our case, we were extraordinarily fortunate, and I'm quite surprised when we explained to the patients as to what this whole project was, and we told them explicitly that there was little or no likelihood of them deriving any benefit from the research we were doing. 95% of all women consented to being in the project. So what it told us was that people actually were being very altruistic. They were not asking us as to what our research would do for them. When we told them that we will help, and all we said was that this research might help us come up with more definitive ways of testing breast cancer for specific treatments in the future, they were more than happy to participate. So I don't think it's ever the challenge of getting them to participate. I think it is the effort necessary for us to explain it to them, and two, I think to be honest, right? If we're honest up front, saying that you're not going to benefit from it, I think most patients who are in that position, vulnerable position, are incredibly magnanimous. That was our discovery.
Fantastic. Do you have anything to add more to the discussion we have had?
In terms of this form of research, so there are two types of research that have received a lot of attention: the fundamental research that happens in research institutes, and then what are called clinical trials. And this sort of research, which is clinical research, with a long-term follow-up and correlating what are called outcomes, that is clinical outcomes, to biological alterations, I think needs to be taken up on a larger basis, not necessarily for breast cancer, but for other forms of disease. And there was some reluctance to do this because of the logistical difficulties and the nature of the project being so long-term. And I think for us the gratifying part is that having succeeded to a measure, in doing this, we recognize that it can actually be expanded not now just to breast cancer, but other forms of cancer, and not merely to cancer, other forms of chronic diseases, be it degenerative diseases, other forms of diseases. Because all diseases have a genetic component and an environmental interaction. Right? So it is the juxtaposition of that particular genetic predisposition and the particular environmental history of that person which leads to the manifestation of disease. And so I think longitudinal studies, cohort studies, help you to explore that in a very interesting manner.